Hello friends, the new topic is the replication procedure in reverse transcribing viruses, that is in case of retrovirus. So the genome of retrovirus is diploid positive sense single stranded RNA. There are two arrows in the different directions and they are the diploid positive sense RNA. First they converted into DNA RNA hybrid molecule. So this is the DNA RNA hybrid molecule by using RNA dependent DNA polymerase. So this is the RNA and this is converted into the DNA. So this is called as RNA dependent DNA polymerase enzyme by using this enzyme. So this all procedure takes place in the nucleus of host cell, and this RNA DNA hybrid is again converted into the double stranded DNA by using the same enzyme RNA dependent DNA polymerase, and this double stranded DNA is inserted into the cellular DNA or host DNA. So the red portion is showing the viral DNA genome, and the blue portion is showing host cellular DNA genome. So this whole genome is called as provirus, and further this provirus transcribes into the mRNA and then spliced. So the viral portion of this provirus is to be spliced and cleaved into the monocystronic mRNA. Then from this monocystronic mRNA the protein is formed. Few of proteins are used as again RNA dependent DNA polymerase, and this is a viral reverse transcribing enzyme. This is not a host cell enzyme, and a few of polycystronic mRNA is converted into again diploid positive sense single stranded RNA genome. So this whole procedure takes place in case of retrovirus replication. Thanks.